Hello, everyone. My name is Sam Mestern, and I'm a PhD student here at Western University, as well as a CONP scholar. And today I'll be talking about my CONP Scholar project, in which I helped facilitate the release of a public dataset, as well as the building of a data dashboard for a dataset based around primate intracellular electrophysiology, or patch clamp electrophysiology specifically. So, neuroscientists have long sought to understand the brain using model organisms. This includes the fruit fly, the squid, and the mouse. All of these have their advantages and disadvantages. However, recently there's been increasing interest in conducting molecular and cellular level characterizations of non-human primate models, such as the macaque, as well as the marmoset not pictured here. My COMP project related to developing an open science database for a collaborative project from multiple labs here at Western involving samples of intracellular electrophysiology and morphology from single neurons in the macaque and marmoset cortex, and then releasing these as a public database. For this project, both macaques and marmosets were used. We received tissue donation from both macaque and marmoset subjects from Dr. Stefan Everling and Dr. Julio Martinez Trujillo labs here at Western University. We then performed patch clamp electrophysiology on the cortical slices retrieved from these subjects. I was fortunate enough to be a patch clamper on this project, as well as working behind the scenes on the database. Using our patch clamp electrophysiology, we performed what we call ePhys fingerprinting, which was a set of standardized protocols meant to characterize the firing and action potentials of the neurons, as well as the subthreshold features. In addition, we filled the cells with biocytin and performed morphological reconstruction of the neurons. Gathering our experimental ePhys fingerprinting, we prepared the data for public release. First, we fed it through quality control and then converted it to the Neurodata Without Borders data format. So why Neurodata Without Borders? We chose this because it's open science oriented. It requires no paid software for usage, and it includes pre-existing tools for analysis in both MATLAB and Python. And finally, it's compatible with pre-existing datasets, most notably the Allen Institute's Neurodata Without Borders data release. So after a conversion to Neurodata Without Borders, we prepared the data for public release, as well as built a browsable web interface, as well as a data dashboard for browsing the cells up close. And I want to dive a bit further into this because this is something I'm really proud of. Here, we built a data dashboard that allows you to browse each individual cell gathered over the course of the experiments. We provide the species, the area, and as well as the pre-computed ePhys features. You're able to expand each cell and look up close at how the cell responded to the long pulse fingerprint protocol that we gave the neurons. And you can view each individual sweep. In addition, we provide filtering tools to allow you to sort by species or by area, and also by ePhys feature. This allows you to really get in depth and explore the different subpopulations of neurons we gathered over the course of the experiment. And I also want to provide a case usage for the Neurodata Without Borders data format that we're releasing the dataset in. Here, I'm going to use the pre-existing Allen Institute IPFX, which is compatible with NWB. It's a Python-based software package for intracellular analysis. Using this package, I was able to perform the Gowans et al. 2019-like feature vector extraction on our NHP data. Here, you're viewing the mouse feature vectors as well as our NHP feature vectors in black as extracted by the IPFX package. From this, I was able to co-cluster our NHP data with the mouse intracellular data as gathered and presented by the Gowans et al. 2019 paper, and you can see our NHP data interlaced with the mouse intracellular data. So with this project comes a number of open science releases beneficial to the CONP community. This includes our data dashboard available at the primatedatabase.com web address, the dataset, which is to be announced, as well as the code for the pipeline presented here, which includes the conversion, recording setup, web interface, and QC code. With that in mind, I want to thank a number of people. That includes the postdoc project leads who did an incredible amount of legwork in order to get this project off the ground. This is Dr. Michael Fairvend, as well as Dr. Eric Kubler. In addition, I want to thank the principal investigators, Dr. Stefan Everling, Dr. Julio Martinez Trujillo, as well as Dr. Wataru Inoue, as well as the database authors, including Will, Kartik, and Dr. Michelle Jimenez, who did an incredible amount of work on the morphological reconstructions part of this project. If you have any questions, feel free to contact me on Twitter, and thank you for watching.